our next session. Thank you so much for returning back. So next, we have our next 4 presenters coming in. The first will be Dr. Diana Vivian, Associate Director of the Division of Bioequivalence II within the Office of Bioequivalence, within the Office of Generic Drugs within CDER. Our next speaker that we'll hear from is Dr. Darby Kozak, Deputy Director of the Office of Generic Drugs within CDER. Next, we will hear from Dr. Jaeyoon Kim, Senior Pharmacologist within the Division of Bioequivalence III, within the Office of Bioequivalence, within the Office of Generic Drugs at CDER. And last, we will hear in this next session from Dr. Ying Fan, Team Lead in the Division of Clinical Review, or DCR, within the Office of Safety and Clinical Evaluation, within the Office of Generic Drugs within CDER. So at this time, if you will help me and welcome Dr. Diana Vivian, I'll let her take the stage.
Thank you for that nice introduction. I'm going to be speaking today on one aspect of the impact of ICH M13A implementation on bioequivalence assessment, and specifically regarding the removal of data due to low exposure. This is a standard disclaimer that this presentation reflects my own views and should not be construed to represent FDA's views or policies. The learning objectives for this talk are 1, to provide an overview of the ICH M13A guidance recommendations regarding the removal of BE study data due to low exposure, and 2, to highlight some case studies showing how OB has implemented these recommendations, including how the scientific principles have been applied for non-immediate release products. So what was our previous practice regarding low exposure data removal? We have 2 FDA guidances that address this. The draft guidances for industry BE studies with pharmacokinetic endpoints for drugs submitted under an ANDA, which was published in August 2021, and also statistical approaches to establishing bioequivalence, published in December 2022. Data should not be removed from BE studies due to identification of statistical-only outliers. And such data was generally only removed along with real-time documentation of a protocol deviation. Otherwise, it could be unclear if the anomalous data resulted from other factors such as product failure. However, in limited cases, an exception was made due to if all of the subject data for one period was below the limit of quantitation and we could not calculate the PK parameters for that period, it disallowed for the use of that subject in statistical analysis, which is a little bit similar to what we're seeing in ICH M13 with some additional caveats. But in that case, it was not possible to calculate PK parameters for the subject without something like imputation, which comes with its own limitations. And it also is likely that in these cases there was a compliance issue and it's not actually due to the product. So you can see here the M13A guidance, bioequivalence for immediate release solid oral dosage forms, was published in October 2024. And I'm going to be speaking mainly about the removal of BE study data due to low exposure, which can be found in section 2B under data analysis for non-replicate study design. This guidance says that generally data should not be removed from the statistical analysis of BE studies solely based on statistical tests that identify extreme values in the PK variables, which is consistent with what you're seeing in the other guidances. There is also language about an exception that can be made for a subject without measurable concentrations or only very low concentrations, and this is the new part in this guidance. Some more detail on this. It could be applicable for either the test or the reference standard product. Specifically, it sets a parameter for AUC for that period. So this could be applicable when AUC for a period is less than 5% of the geometric mean AUC for the drug product overall. And the geometric mean AUC should be calculated without including data from the low exposure subject. Because this is considered to be the result of subject noncompliance, exclusion will only be accepted in exceptional cases, in general, with no more than one subject in each study. And of course, applicants should proactively minimize the potential for subject noncompliance in the conduct of the BE study through their study design and protocol, for example, by mouth checks after dosing to make sure that this is minimized as much as possible. And even if an applicant removes subject data because of low exposure, the data should still be submitted for agency assessment. Now I'm going to get into some case studies of how we've implemented this in OB. The first case is for an immediate release product with a two-way crossover BE study. And you can see the individual subject data profile shown here, where period 2 of the reference product is much lower than this subject's period 1 data of the test product. Though after reference standard treatment for this subject, the observed AUCT was 3.58% of the geometric mean AUCT for the reference standard overall. And like I mentioned, this is excluding the low exposure subject for that calculation. When including the subject, both Cmax and AUCT failed to meet BE criteria. You can see at the higher end of the confidence interval there, they fail. When excluding this subject, it brings the confidence interval dramatically differently into range. For this case, the removal of the low exposure data was considered acceptable because it met the scientific principles outlined in ICH M13A, including that the AUCT was less than 5% of the geometric mean AUCT for that product. And when the low exposure data is included, you could see that the TR ratio for AUCT, AUCI, and Cmax were close to 1, and the 90% confidence intervals were all within BE limits. And further, based on available data, such low concentration is not considered to be typical of the reference standard. Case 2 was for an IR product with a fully replicated BE study design. In this case, a reference scaled BE approach was adopted due to high variability. In the individual subject data here, you can see the period that the applicant wanted to exclude was that shown in red, the lowest reference product treatment. It's not as obvious as the previous case, where it's actually pretty close to the period 1 reference product data also, and we'll get into that. The AUCT of that period compared with the geometric mean AUCT overall was 7.6%. And the AUCT and Cmax results failed to meet acceptance criteria using the reference scaled approach when this subject was included, but it met if excluding the subject. However, in this case, it was not acceptable, for one, because the AUCT was greater than 5% of the geometric AUCT of this product in the study, and also due to the highly variable nature of the drug product, similarly low concentrations were observed both within this ANDA and across other ANDAs, and were not excluded in other cases from statistical analysis. And it really suggests that the observed low exposure is not likely due to subject noncompliance. So this addition in ICH M13A is meant to account for really realistic subject noncompliance that we've seen over the years, but it's not meant to just be a statistical outlier for something that may be due to product performance. And the applicant in this case was recommended to repeat the study. Case 3, although ICH M13A is not for modified release products, we'll show how it was used in this case for, using the scientific principles. This is a modified release product with a two-way crossover BE study. I think you can see in this case, I think there were only 5 measurable concentrations for period 2 of this subject, which was given the test product, compared with a more typical profile for the reference product. The AUCT was 1.57% of the geometric mean AUCT for the test product, and including this subject, AUCT, AUCI, and Cmax failed to meet BE criteria on the lower end. So although M13A is not applicable generally for MR products, we used a risk-based approach using the scientific principles outlined in the guidance and some other risk considerations you can see here to evaluate this. For one, the AUCT in this case was less than 5% of the geometric mean AUCT. The concentrations we observed were very close to the LOQ. Such low concentration was not expected based on the overall variability of the product. Excluding the subject, the TR ratios for AUCT, AUCI, and Cmax are very close to 1. And we looked at some other risk of the product overall, including that there was a similar formulation between the test and reference standard products with the same type and grade of release controlling excipients. And there were comparable dissolution profiles between the test and reference standard product in QC media and also multi-pH media. And further, this product is meant to be taken once daily with an evening meal, suggesting the already acceptable fed BE study has more relevance potentially than this study. So this was all taken together as a totality of evidence approach to conclude that this was likely due to subject noncompliance and could be excluded. And the last case is for a narrow therapeutic index drug product, MR drug product with a fully replicated BE study design. In this case, the replicated BE study design was due to the NTI nature of the drug and not due to high variability. So for an NTI drug, the acceptance criteria include 95% upper confidence bound from reference scaled BE less or equal to 0, unscaled 90% CI within 80 to 125%. An upper limit of 90% equal tails less than or equal to 2.5. And you can see the profile here. The AUCT compared with the geometric mean AUCT overall was extremely low, only 0.27% for this one period, period 4 after test treatment. You're only seeing 2 periods here, even though it's a replicated BE study design because the subject withdrew from the remaining 2 periods. So we only had 2 periods of data to work with for this subject. Including the period 4 PK data, the AUCT, AUCI, and Cmax failed to meet acceptance criteria from unscaled BE assessment, but still met the reference scaled BE criteria. And again, similar to case 3, we used a totality of evidence risk-based approach to evaluate whether this it was likely due to subject noncompliance or could be something to do with the product. In this case, the AUCT was less than 5% of the geometric mean AUCT of that product. The 90% CI, when excluding the subject, were in a very tight range of 96 to 102%. I know we don't have an official narrow range, but it does seem very close to 100. And the observed plasma concentrations were all very close to the LLOQ of 0.05 microgram per ml. Further, we looked at the formulation and release mechanism of the test product, and it was very similar to the reference standard product with the same type and similar grade of release controlling excipients. Now I'll get into challenge questions. I think this one should be an easy one. Which FDA guidance document contains information about an exception for removal of BE study data due to low exposure? A, BE studies with PK endpoints for drugs submitted under an ANDA, published in August 2021. B, statistical approaches to establishing bioequivalence. C, M13A bioequivalence for immediate release solid oral dosage forms. Or D, M10 bioanalytical method validation and study sample analysis. I hope if you saw the title, you know the answer is C. And for challenge question 2, which of the following statements is not true? A, low exposure data removal may be applicable if AUC for that period is less than 5% of the geometric mean AUC for the drug product without inclusion of the data from the low exposure subject. B, data removal due to subject noncompliance may be applicable for the reference standard product only. C, exclusion will only be accepted in exceptional cases, in general with no more than 1 subject in each study. And D, all data, including any data excluded from statistical analysis for low exposure, should be submitted for assessment. And as we saw in the case studies, data removal due to subject noncompliance could be applicable for either reference standard or test product. So it's not limited to the reference only. In summary, the M13A guidance, bioequivalence for immediate release solid oral dosage forms, introduced scientific principles for the exclusion of low exposure BE study data, which is considered to be due to subject noncompliance. Removal of low exposure data will only be accepted in exceptional cases. For example, if more than one subject in a study shows low exposure, this could bring other questions of dose administration reliability or product failure. And although M13A applies for IR products, the scientific principles of low exposure data could be considered for other types of products, as you saw in the 2 case studies that I mentioned, using a risk-based approach and totality of evidence to assess whether the low exposure data is in fact likely due to subject noncompliance or if there are other questions it raises. And I'd like to acknowledge the 4 folks here, especially Ming and Lucy, who helped me with a lot of the data analysis. Oh, and I'd like to welcome Dr. Darby Kozak.